As you can probably see from my talk, we are obsessed with platelets. So what are platelets? They're actually not cells because they don't have a nucleus. They're derived from megakaryocyte in the bone marrow, as I would show in the next slide. They're highly abundant, so they outnumber most of the other cells in the blood, have a lifespan of 7 to 10 days in circulation. And apart from their classic role in thrombosis and hemostasis, they're now recognized as potent inflammatory cells as well. And I just put this, oops, sorry. Maybe I should stand here. I put this out from this really nice Cell paper from 2017, where this group could show that they actually can migrate as well, so similar to monocytes. So you see this platelet here, when you throw something which produces a chemotactic gradient, the platelet will actually move towards this. And it's highly dependent on the cytoskeletal arrangement. So if you look at this platelet here, it's moving, and then you put a blocker of the cytoskeleton arrangement in, it basically gets stopped in its tracks immediately. And also I find this quite fascinating here. These are platelets, when you expose them to bacteria, they basically grab the bacteria and collect them and remove them in certain circumstances. So they're not only important in thrombosis or inflammation, but they're really highly versatile cells. And they undergo a dramatic shape change, as you can see here from this picture. It's a normal discoid form, and when they're activated, they undergo this dramatic shape change. So where are they coming from? They're coming from megakaryocytes, as I mentioned. So they're basically just membrane blobs. So you see that they're kind of these little things which come out of the megakaryocyte over time. These are the platelets, the little top things. So why we are interested in platelets? They play a role in a lot of diseases. Cardiovascular disease is the obvious thing, but they also play a role now in cancer. They have been attributed for some neurological diseases like MS and also some neurodegenerative diseases. And this is just an overview how these diseases develop over time in the different brackets of income per capita. So these are the wealthy countries. That's middle income, that's low income. And you see that especially in the middle and low income, cardiovascular disease is clearly on the rise. And that's why you're interested in platelets for the diagnosis and therapy as well. So from the diagnostic point of view, what's important, you know, unstable plaques causes myocardial infarctions and strokes. And it's really a nasty disease in a way, because the plaques you can usually see on an angiogram when you just inject contrast. These are plaques which are highly stenotic, and they're really responsible for a low number of myocardial infarctions or angina. The plaques which are really dangerous and they cause the myocardial infarction, they have this outward modeling, and you can't really see this because the lumen is fully intact on a normal angiogram. So we're really working for a number of years now to develop imaging agents which can detect the inflammation in these plaques, in these highly dangerous plaques. And we think that platelets are a very good target for that. So we're looking at a receptor on platelets which is highly abundant. This is the integrin GP2B3A, which is the fibrinogen receptor. It's specific for platelets. It's highly abundant, as I mentioned, which is great for imaging. And also it undergoes a very dramatic shape change when the platelet is activated. So it goes from this bent form shown here to this open form when it attaches then fibrinogen. And because that's the non-activated and that's the activated form, you can potentially make an agent which only detects the activated form. Unfortunately, all the clinical approved drugs recognize both forms of the receptor. So they're really not detecting activated platelets specifically. And for imaging, that's of course not possible because you would image all platelets in the circulation. So there's one antibody against 2B3A out there, as I said, a non-activation specific antibody, which is a Fab fragment called ReoPro. And this is just an overview of the antibodies which are currently under development clinically. Mainly these are IgGs, so it's probably the mainstream antibody, but fragments now get more traction with the approval of some of the nanobodies as well. We are specialized in single-chain antibodies, which are shown here, which are a fusion of the light and the heavy chain of a full IgG antibody. For imaging, they have lots of advantages because they're very small, so they clear rapidly, so you don't get a lot of background when you do diagnostic imaging. So how do we make these antibodies? Most of our antibodies we make these days are made by phage display. This technology got a lot of recognition last year because it was awarded the Nobel Prize for George Smith and also Greg Winter for phage display. And Monash also has recognized the importance of phage display, and they're actually putting some money towards an in-house library which we can use without strings attached comes to IP, and we're also trying to make a very good library. So there are commercial libraries out there, but they're all not very good, and you also have strings attached when it comes to IP and commercialization. So that's under development, and hopefully by the end of the year we have some in-house library available. So I go a little bit into the process of phage display. So you have bacteriophages, which are shown here, which are basically a virus for bacteria. So you can infect bacteria with this. And you have the classic combination of the phenotype and the genotype. And that's very important because your antibody has to be linked to the genetic information which encodes this antibody. And you have millions and millions of these phages encoded on a plasmid here. And then you can go through selection rounds and isolate specific binders for your target of interest. And that's for a number of years ago was done for this 2B3A target. So briefly to describe the strategy, you basically take non-activated platelets, you incubate with your library, you delete all the unspecific binders and you throw them out, then you activate your platelets, you do another selection. So what you end up after 4 to 6 rounds is really binders which are not binding to the unspecific non-activated cell surface or the unactivated receptor, but specifically bind to your activated receptor. And that's what finally came out, the first activation-specific single-chain antibody against activated platelets. And here in the flow cytometry diagram, you see that the antibody is binding specifically to activated platelets, not to non-activated platelets, compared to the clinically approved drug Acipsimab. So that also works in vivo quite well. So the first experiments we did in the early days was for MRI. So we used iron oxide particles. We conjugate these iron oxide particles to this antibody. And in a model of mesenteric injury of the mouse, you can see that these particles which have a high autofluorescence bind very specifically and rapidly to this developing thrombus. That is also important for plaque imaging, was found with these human samples. So these are samples the surgeon took out of the carotid artery of someone who had a stroke or some transient attacks. And here you can see that these iron oxide particles stick to the surface of these vulnerable plaques, and they're co-localized with the red staining of the platelets. And here you can see the MRI scans, so that's the targeted agents before and after, and this is a non-targeted agent. So you can, if the light would be a bit darker, see that there's more black on the surface of these plaques compared to before or compared to the control. But you would also appreciate that MRI has an inherently low contrast. So MRI is a very low sensitivity modality when it comes to T1 and T2 contrast. And realizing that, we rapidly, after our first MRI experiments, moved into PET, because PET with radio tracers has a much, much higher affinity and specificity and also sensitivity. So we have one of the most productive PET facilities in the country, I think, driven mainly by my former postdoc and now lab head at ACBD, Karen here. So that's literally was right below you, underneath this lecture theater, was the situation of the old PET scanner. And that's actually the old PET scanner here. And we have also the associated radiopharmacy handling facilities to handle some of these isotopes shown here, gallium, fluorine, and mainly we do copper. I'll come to that a bit later. Thanks to the generous support of the university, this PET scanner was replaced by a new version late last year, which has a much more powerful CT, a micro CT, and also has a 4-animal bed where you can image 4 animals at the same time. So you get much higher throughput with short-lived isotopes as well. So we then attached this antibody to a radio tracer. In our case, we used copper. That's a collaboration with Paul Donnelly at Melbourne University and also Brad Patterson, who was a postdoc in Paul's lab and is now heading the radiopharmacy at Monash. So he's based in Clayton and is driving all the radiopharmacy there. So Paul and Brad developed this MECOSAR cage, which is a specific cage for copper ions, and they can trap copper very efficiently without leaking off in vivo. And by conjugating this to the antibody and injecting it into a mouse model of carotid artery injury, you can see here that you get a nice accumulation of this radio tracer in the mouse injury. And with PET, of course, you can do also dynamic longitudinal imaging. So you can put the mouse in the scanner, you can inject your radio tracer and follow the accumulation of the tracer over time, which is shown here. So you see that when the tracer hits the bloodstream, it accumulates roughly around half an hour it takes to have the maximum saturation of this tracer in the mouse. And it's also cleared after half an hour. So that's an ideal agent because you get really a lot of background reduction with this fast clearance and high accumulation. So what's the clinical translation? After we published these papers on this radio tracer, Clarity basically licensed the IP from the Baker Institute, where originally this technology was developed, and they got funding from the European Union to basically develop that into a clinical agent. And maybe in a few years, who knows, you can go to the hospital and say, I would like to have a plate view examination, which will check your plaques and your risk of getting a stroke or a myocardial infarction. We are in parallel working on short-lived isotopes. So the issue with copper is a great isotope, don't get me wrong, but it has a very long half-life of 12 hours, which for an antibody which clears within half an hour is a bit of an overkill. So we're trying to look at shorter-lived isotopes, which are also more readily available. And gallium is our top candidate. So copper you have to make on a cyclotron, which is shown here basically. These are these multi-million-dollar machines, which you have to have in a bunker pretty much to make copper radioisotopes. With gallium, you basically buy this benchtop generator, which you can milk a couple of times a day, and you can get multiple doses of gallium from a long-lived isotope, germanium. And it's very versatile. You can have that in each hospital to make your radio tracer basically in situ. The problem with that was that the old chelators shown here, they all require a very high temperature to capture the gallium ion in this cage. So when I was presenting this work a couple of years ago at a conference, a nuclear medicine conference, Frank Roche came to me after the talk. He's here from Mainz. He's one of the pioneers of gallium chemistry. He actually invented these gallium generators. So he has a strong track record in gallium chemistry. And he said, why do you use copper? Why do you not use gallium? I have this really interesting chelator called DATA, shown here, which can bind gallium at room temperature within minutes. So our very talented PhD student Jackie, shown here, embarked on this project where she basically used this DATA chelator, attached it to the antibody, and this is fresh off the press in a way, unpublished data around proof that we can now label this antibody with gallium and have a much higher availability potentially in the hospital with these generator-based isotopes. So I'm shifting, the second half of my talk, shifting gear a little bit into therapy. So if you can't diagnose your vulnerable plaque because the tracer is not available at the moment, you need to treat the patient very fast and efficiently. And this is a very old paper, but I still like that very much because it shows dramatically after myocardial infarction what your therapy options are and what the long-term survival is. And it shows that if you act early and you actually do pre-hospital thrombolysis on top of primary PCI, your survival rates are much higher in the long term. And that's true for stroke, but also for myocardial infarction. And I'm always surprised, I'm not sure how much they have now in Melbourne of these mobile stroke units. There might be 5 or 10 now, but you see them quite often if you go to the city. Have a look if you come across some of these. They have a built-in CT in the ambulance where they can exclude a hemorrhagic stroke straight away and then basically give thrombolysis in the ambulance before the patient even reaches the hospital. Another interesting story, which you might have seen here in the New England Journal, was this 44-year-old nurse which was out in a paddock somewhere in Western Australia, and he had a myocardial infarction, and he realized that he had a myocardial infarction. He put his ECG leads on, he sent the ECG to the next hospital and got the confirmation that he had an MI, and he basically thrombolyzed himself. So he injected the drug himself and survived. And of course, he was trained, he was a nurse. I wouldn't recommend that for everyone, but it's definitely showing very, very dramatically what the chances are, what the risks are if you're far away from hospitals when you have an acute MI or an acute stroke. So a safe therapy for thrombolysis would be something highly desirable. And we are doing that in the setting of nanotechnology. Not sure why that's also distorted, but basically nanotechnology are small, smart delivery vehicles. And we are working with the University of Melbourne again, together with Frank Caruso, who is shown here. And we make layer by layer or self-assembled drug vehicles. So you put your drug in a mesoporous template, and then you put polymers or some coating around, which can be degraded by something what you embed into this coating. And by doing that, you shield the drug from the bloodstream, and you also can release that pretty much on demand. So the principle is shown here. You have your clot building up, and then you have these little smart nanoparticles with the antibody on top rushing towards the occlusive clot and then bursting at the site of the clot and releasing the drug to treat the patient. And we thought that we might use the clotting cascade to release the drug. So we came up with this idea to use thrombin as a trigger to release the drug from these particles. And just to remind you how important thrombin is in this setting, just put the coagulation cascade here, and you see that thrombin is central for all these processes leading to acute thrombosis and clotting. And you can basically hijack the coagulation system in this sense to remove a clot. So as I said, we made this little smart vehicles where we load UPA. We coat on both sides multiple times. We put an antibody on the surface. And this is the sequence which is important. So this is the sequence which is cleaved by thrombin. And we have a control sequence as well. So once these particles see thrombin, this is cleaved and the entire particle falls apart. So this is just some characterization of this particle. So we can see the layers building up. And also we showed that they're binding to activated platelets. And the most important part was really looking at the degradability and the way these particles can fall apart. So these are all the controls over time. And now you see if you add, not sure what's going on. If you add some thrombin, the particles start to degrade. And then if you add a little bit more, they start to degrade faster. Still, it wasn't that fast as we would hope for, but that's 2.5 units of thrombin. So if you go higher to 2 or 2.5 or to 5 units, the release of the drug is accelerated. And if you finally come up to 10 units of thrombin, you literally see that these particles disintegrate within seconds. And that's really what you want in an acute setting. What also was important for us is that if you have the antibody on the surface or not, it doesn't really matter for the degradability. Because you can imagine if you have a nanoparticle and you have antibodies on the surface, the thrombin might have difficulties to reach the polymer lying underneath and come past the single chain. But to our surprise and very pleasantly, we saw that that's not an issue. So the thrombin can still degrade the particles quite efficiently. And that's a current NHMRC grant we have since last year to further develop this system and test that in vivo as well. So I hope in summary I have convinced you that activated platelets are a highly attractive target for diagnosis and therapy of thrombosis and inflammation. And we have developed a novel targeted radiotracer for the detection of activated platelets, which is moving towards the clinical use thanks to Clarity, who has licensed this IP. The use of gallium is an isotope which could probably speed up translation of this radio tracer by reducing the cost and increasing the availability. Targeted and clot-busting nanoparticles could lead the way out of the current stagnation in the field of thrombolysis by reducing adverse effects. So that leaves me to thank all the funding bodies, NHMRC, Heart Foundation, and ARC, Clarity, and Mediso, Baker collaborators in Germany, the Austin for providing the copper from their cyclotron, Frank in Mainz, Paul Donnelly, and they're part of this ARC Center of Excellence. You can follow us on social media if you like, and thank you for your attention.